Hello again. We continue in our document 1 and we arrive to the HIV cycle, part 3. So, HIV attacks cells carrying the CD4 molecule, which is a marker for T helper or LT4 and also macrophages. So, HIV binds to its target cell, which is mainly T helper cell, via the glycoprotein 120 expressed on its surface. The glycoprotein GP120 binds to the CD4 protein expressed on the surface of T lymphocyte. So the fixation of HIV to its target cell is ensured by complementarity between the viral protein GP120 and the membrane protein CD4. This fixation permits the fusion of the viral membrane and the cytoplasmic membrane of T helper, allowing the entry of the virus into the cytoplasm of the target cell. So here a diagram summarizing the HIV cycle. First step is the binding of the HIV virus to its host cell through the complementarity between the glycoprotein of the virus and the CD4 molecule. This binding allows the fusion of the viral membrane with the host cell membrane, which allows the entry of the capsid containing the HIV RNAs, the reverse transcriptase, integrase, and other viral proteins within the host cell or the cytoplasm of the host cell. So here we have the penetration of the capsid containing the viral genome and the associated proteins in the cytoplasm. Now the capsid disintegrates and releases the viral RNA and the reverse transcriptase. Now the RNA will be transcribed into viral DNA, it's the third step, by reverse transcription. The obtained double-stranded viral DNA is transported in step 4 across the nucleus and integrates into the host DNA. So as you can see here, in yellow is the viral DNA reverse transcribed, and in blue it's the host cell DNA. So we have integration of the HIV DNA into the T4 DNA. Now the T helper will transcribe its own DNA and the new viral DNA. We obtain messenger RNAs, which are viral RNAs that leaves the nucleus. The viral RNA leaves the nucleus, enters the cytoplasm, and makes viral proteins. So the viral RNA is translated into viral proteins in step 5. Now the new viral RNA and the formed viral proteins move to the cell surface in order to make new viruses or new HIV forms. Thus, in step 7, the virus is formed again, and the new viruses, the mature variant or the new viruses obtained, are now released from the host cell, and they are ready to infect other target cells, other T helpers. So these are the steps of the HIV cycle. You don't have to memorize the steps, but only to understand the mechanism by which HIV infects the host cells, mainly the T helper. In this slide, we have a video that summarizes the steps of HIV cycle. So here the structure of the human immunodeficiency virus. We have the lipid envelope, and we have the glycoproteins 41, which is a transmembrane glycoprotein, and we have the glycoprotein 120. We have the capsid here that protects and surrounds the viral RNAs. The RNAs are associated to proteins, mainly the reverse transcriptase, and we have also protease and integrase. Now, the virus binds to CD4 molecule via its glycoprotein 120. CD4 is expressed on the surface of T helper. This binding allows the fusion of both viral membrane and T helper membranes and the release of the viral genome in the cytoplasm. The RNAs and the reverse transcriptase are used to make viral DNA, as you can see here. The viral DNA now, the double-stranded DNA, is ready to move towards the nucleus and to be integrated within the DNA of the host cell. So here the viral DNA is integrated using the integrase enzyme into the DNA of the host cell. Now the DNA will be transcribed into mRNA using the tools of the T helper. The RNA, the viral RNA, leaves the nucleus, enters the cytoplasm to form proteins, viral proteins, and now the RNAs and the viral proteins associate together to form new viruses ready to infect other T helper cells. So in this way, the HIV infects the host cell. In the next video, we are going to see the progression of the infection by HIV virus as well as the possible treatments. Thanks for listening.